The discovery of the structure of DNA had clear implications for how DNA could be replicated or copied during cell division, because the base pairing allows one strand to serve as a template or as a sort of a master copy for a new strand to be made. However, there were doubts about whether the mechanism or method of copying DNA is conservative or semi-conservative. Now, in conservative replication, and this is a hypothesis, is that let's say we have a helical DNA like this, and in order to copy, the two strands will separate, and then we will make two new strands. And this would then result in two double helices. Both the daughter strands would pair up, and both the template master copy or mother strands would pair up. This is conservative replication. In semi-conservative replication, you have the double helix. The double helix splits up and is used as a master copy or template using base pairing to make two new daughter strands. However, they don't the new strands do not combine with each other to make a double helix, but they just pair up with the parental strand. So the daughter molecules or the new double helices look like this. And the experiment that confirmed that DNA replication is semi-conservative was carried out by Messelsohn and Stahl. And they used a technique called cesium chloride gradient centrifugation, in which you have a solution of a salt of cesium chloride, and you spin the tube of cesium chloride very fast for a long amount of time. And what that does is that you form a gradient of cesium chloride density where there is very dense cesium chloride at the bottom of the tube because of the centrifugal force being applied in the downward direction if you are spinning the tube like this around this point as a center. So because of the centrifugal force, most of the cesium chloride ions are going to go to the bottom, and there will be a very high density of cesium chloride at the bottom, and there will be a very low density of cesium chloride at the top. And what this means is that any molecule, let's say DNA, will float to the point in this tube that matches its density. So if the DNA is in the high density area, and the density of cesium chloride is higher than the density of DNA, then the DNA is going to float towards the top. On the other hand, if the DNA is in an area of low density, so that the density of DNA is more than the density of cesium chloride, then the DNA is going to sink towards the bottom, and it'll come to rest at the position in the tube where the density of the cesium chloride matches the DNA's density. And therefore, this allows you to separate different types of DNA based on their density. Now, there was another trick that Messelson-Stahl used, and that trick was they used two isotopes of nitrogen. 14N is the most abundant isotope of nitrogen, and it has 7 protons and 7 neutrons. However, rarely you can also find 15N, which has 7 protons and 8 neutrons. Since the number of protons is the same between the two isotopes, their number of electrons are also the same, and therefore the chemical properties of the two isotopes are identical. The only difference between the two isotopes is that 15N is heavier than 14N. So what Messelsohn and Stahl did was they grew E. coli in medium that contained the heavier isotope of nitrogen, and they grew them for a while like this, so that all the DNA, all the nitrogenous bases that contain nitrogen now had 15N, the heavier isotope of nitrogen. Once they made these E. coli whose DNA was made up of this heavier isotope of nitrogen, they shifted these E. coli into medium that now contains the more common isotope of nitrogen, which has an atomic mass of 14. And they allowed these bacteria to undergo one round of cell replication. And if conservative replication was happening, what you would have seen is a heavier, some molecules would be heavier and some molecules would be lighter. Whereas if semi-conservative replication were true, you would see molecules having a density that's intermediate, which is shown here as well. And then when they actually carried out the experiment, they observed that the prediction based on semi-conservative replication was true, and all the DNA after one cell division had intermediate density than the 15N DNA or the 14N DNA. And then if you carry this experiment forward one extra cell division, then what you expect is that when this molecule replicates, you're going to have one template strand that's made up of 15N, so it's heavier, and the other template strand that's made up of 14N, so it's lighter. And since the medium now contains 14N, you will have both daughter strands be made up of the lighter isotope of nitrogen, 14N. And the same thing will happen to this other strand, where you will have one template which is the DNA from the original E. coli that had been grown in 15N, and the other mother strand or the template strand will be made of 14N. And then when you copy these two, you will get two double-stranded DNA molecules, this one and this one, also shown here as this one and this one, which are of intermediate density, but you will end up creating two new double-stranded DNA molecules that are entirely made up of 14N, and therefore when you do the density centrifugation, you should get a new band which is of has a lighter density, lower density than either the density of 15N or the intermediate density. And when Messelsohn and Stahl carried out this experiment and carried the E. coli forward for one extra cell division into the second generation, that's exactly what they observed. And the experiment is a landmark experiment that confirmed that DNA replication is semi-conservative and not conservative. Next, let us consider the process of DNA replication in more biochemical mechanistic detail. Now, the reaction that makes a new daughter strand is a polymerization reaction. Since the DNA is a string of nucleotides connected together, so it's a molecule that's made up of repeating units, DNA is basically a polymer. And the process of making this polymer longer or bigger is polymerization. This reaction is not spontaneous. You cannot spontaneously grow the daughter strand of DNA, and you in fact need an enzyme. And recall that an enzyme is a protein that can catalyze reactions. So here you have an enzyme that has the ability to create to add nucleotides to the growing chain of the daughter nucleus, the daughter strand, and that enzyme is called DNA polymerase, since that enzyme's job is to polymerize DNA. And so let's say we have a template strand that we are trying to copy, and in this particular strand, the fifth carbon is over here, the third is over here, so that the fifth of the next nucleotide is over here, and so on. And what that means is that on the other strand, since the strands are anti-parallel, 5' is going to be here and 3' is going to be here. Now let's further assume that we have added nucleotides up to this point, and our job is going to be to add a new nucleotide at this guanine in the mother strand or the template strand. So the first thing that's going to happen is that a cytosine nucleotide is going to come and base pair with the guanine. Now, the molecule that comes and base pairs is called a deoxynucleoside triphosphate. And it's so called because a deoxynucleoside is the ribose sugar along with the nitrogenous base is the nucleoside. And instead of having just one phosphate group, there are three phosphate groups, or a triphosphate that's attached to the fifth carbon. And so the raw material for polymerization are deoxynucleoside triphosphates, or dNTPs. And the reason we need a triphosphate and a monophosphate or a diphosphate will not work is that these phosphates are negatively charged, and you need the high energy of these negative charges in order to carry out the polymerization reaction. In the next step, DNA polymerase catalyzes the formation of this ester bond and causes the release of these two phosphates as pyrophosphate also released is water, and that's why this is a condensation reaction. And finally, this is an exergonic reaction since energy is released. So even though we need the high energy of the negatively charged triphosphate in order to initiate the reaction, in the end the energy the reaction releases energy and is an exergonic reaction. And this leads to the formation of the ester bond between the 3' carbon of the pre-existing nucleotide with the 5' carbon of the new nucleotide. An important consequence of having the requirement of a triphosphate and having the requirement that there must be a hydroxyl group on the 3rd carbon in order to carry out the reaction is that you can only add bases to the 3' end of a growing daughter strand. And that's why we say that DNA synthesis occurs in the 5' to 3' direction. However, it's important to keep in mind that when we say DNA synthesis is 5' to 3', we are referring to the daughter strand. From the perspective of the parent strand or the template strand, the DNA synthesis is in fact occurring in a 3' to 5' direction. So if you say that DNA synthesis is occurring 5' to 3', you are always talking about the daughter strand. And in reality, all that means is that you can only add new bases, new nucleotides to the 3' end of the daughter strand where a hydroxyl group is available for the triphosphates to react with and make the ester bond. One last thing, and a rather important thing worth pointing out is that the requirement that there be a hydroxyl group here on the 3' carbon in order for the triphosphate to react and make the phosphodiester bond, means that you must have a pre-existing molecule that makes that hydroxyl group available. Now, when the chain is growing, that's easy to see that the nucleotide that was previously added to the growing chain or polymerized will provide that hydroxyl group. But what about the situation at the beginning of DNA replication, at the start of DNA replication, before any new nucleotides have been added? For instance, let's say we have a template strand that looks like this, and we have to start synthesis. However, there is no nucleotide that has a hydroxyl group on the 3rd carbon available in order for the triphosphate to have the reaction and form the phosphodiester bond. So how will synthesis or polymerization get initiated? And the need to have some sequence that provides a hydroxyl group, that means a sequence that starts the process off or primes the process, means that we need what's known as a primer. And a primer is just a complementary sequence of nucleotides that provides a hydroxyl group at the 3' end in order for us to initiate or start the process of polymerization or building the daughter strand. And you will note that here I said U instead of T, since this is an RNA primer. So the process of DNA replication requires that we have a primer that's complementary to some part of the template strand, and that provides a hydroxyl group to which the first base can be added through the polymerization reaction. And after that, each new base that's added will provide the hydroxyl group, so that you keep adding new bases to the 3' end of the daughter strand.